Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConell. I've just finished chatting with Professor Paul Greenhaff from the University of Nottingham. He's been an expert in exercise metabolism for many, many years, looking at all sorts of things, including creatine metabolism, fat metabolism, carbohydrate metabolism. And recently, he's been looking at the metabolic effects of inactivity. And interestingly, he has a theory that inactivity is not simply the opposite of activity from a metabolic standpoint. So he's been finding with an inactivity model that he uses, which is immobilization. So you either immobilize the arm or looking at bed rest, he finds that you get a reduction in your insulin sensitivity. So your insulin stimulated glucose uptake within 24 hours. So if you think, you've been laid up, if you've injured or you're sick in bed, within 24 hours, this inactivity will reduce your insulin sensitivity. This is important because inactivity is a major risk factor for mortality, and the mechanisms aren't that clear. So he's finding here by doing well-controlled studies that you get a reduction in insulin sensitivity, and we know that that can be a cause of pre-diabetes and diabetes. And also this reduction in insulin sensitivity increases your risk of heart disease and even cancer. It's important work, so stick around. I think you'll really enjoy this one. Hi, Paul. Welcome to Inside Exercise. Thanks for coming on.
Hi, Glenn. Thank you for the invite. It's always nice to chat to you.
Yeah, I was thinking a couple of days ago that I had Javier Gonzalez on here talking about carbohydrate metabolism, and I told him I'd only ever been to London. And then I realized later, it's like, hang on, I stayed at Paul Greenhaff's house in Nottingham like 20 years ago or something, and Oscar Hugen's house as well in Birmingham. So anyway, thanks for letting me stay at your house all that time ago.
It's probably nearer 30 years, but never mind.
No, you're right. It's 30 years. It was creatine. It was when we were both doing creatine. Well, you were the main creatine guy. Anyway, thanks for that. And he also said that there was more canals, or about the same number of canals in Nottingham as there is in Venice, which kind of blew me away.
Well, there's a lot of canals in Nottingham. I know that. I've never counted them in Venice, but yeah, for sure.
Well, that's interesting anyway. All right.
Good for cycling.
Good for cycling. Oh, nice.
Yeah.
All right. So we're going to be talking about physical activity, exercise, etc. But what I often do when I start is I sort of wonder, do people, get into exercise research because they were interested in sport and exercise initially, or were they like a scientist to then, quite a lot of people have sort of come the sport and exercise route. How did you get into all this research?
A bit of both, really, Glenn. So my undergraduate degree was a joint honours degree in physiology and human performance. So after I completed that, I knew I wanted to delve more into exercise physiology, really. So I went to Aberdeen to do my PhD with Ron Morn, and I've been very lucky to have some, fantastic mentors. Ron, Roger Harris, when I was working on thoroughbred racehorse muscle, and then Eric Hultman, of course. So I'm really lucky, and very lucky to have some great students and collaborators in my 30 years in Nottingham too. So
Absolutely.
Yeah, very lucky. I mean, science is as much luck and being in the right place at the right time as it is hard work, of course.
Exactly. All right. So you've done well, obviously. And those are a couple of those people are actually on my list of potential podcast guests. So we'll see what happens. Now, because you're going to be talking about inactivity, the imperceived muscle stressor, how about we just sort of set the scene a little bit? So we're going to be talking about inactivity and getting to some of the, the ways you've looked at that. Why don't we just talk a little bit about, what is physical activity? What is inactivity? What are different ways of looking at that? And then, without getting into too much detail, we'll start sort of moving towards what you've done and why. Yeah.
Yeah. I mean, most of the, so if you have high, moderate and low physical activity levels, that's what you generally see in the literature. Those are mainly based on step counts and mainly based upon epidemiology research, and epidemiology research into health. So, large scale longitudinal studies or cross-sectional studies where they've looked at physical activity levels and related that to to either cardiometabolic health or mortality rates. So there's that sort of research. Then there's also chronic disease states where physical activity levels are very low. So, you know, like COPD, where step counts around about 3,000, 2,000 steps per day. So I guess what I'm saying is the step count is the usual way it's measured. General guidelines are, 10,000 steps per day is a high activity, 7,500 to 10,000 is moderate, 5,000 to 7,000 is low, and below 5,000 is sedentary. But I must stress, I guess, that the biological mechanisms underpinning this are not yet resolved. But the epidemiological evidence is pretty convincing.
Yep. So I guess people have started doing studies with, I mean, it gets very, very hazy, doesn't it, this inactivity? Because as you said, I suppose you could have 5,000 steps, but it might be like all of it might be going upstairs. I mean, I know I'm just going, kind of the extreme. And then you might have someone else that's doing 10,000, but they do it all sort of, before work, and then they sit around for 12 hours or whatever. Yeah. So,
I mean, there's a whole debate around, the impact of sedentary activity as opposed to low step count, the frequency of activity doses, if you like. There's a whole debate around that, but it's primarily based upon fairly imprecise measures. Not many studies have measured metabolic rate, for example, over 24 hours and tried to match this with different physical activity levels. So yeah, the epidemiology is quite interesting. There's a lot of nice research where 50,000, 80,000, 1 million volunteers, where they've shown that high levels of physical activity can have a major blunting effect upon all-cause mortality, for example. The mechanistic basis of that, we don't know. For me, though, where I'm trying to focus my interest is trying to understand more about inactivity. People talk about physical inactivity and they study it by introducing activity. What I'm endeavoring to do is try to understand more about the biological mechanisms that are causing adaptation in muscle during inactivity. And I think if you know that, if you can understand the biological basis of the impact of inactivity, you can then design studies focused on how to alleviate that. And of course, inactivity will affect more than just muscle. So cardiac tissue, even the brain, there's a relationship between physical activity levels and gray matter volume of the brain. So it's really interesting. And the idea that being inactive doesn't really do a great deal compared to physical activity or exercise that does a lot of positive things, I think is probably incorrect, in that it sort of fits with the title that I gave you, that inactivity in itself is a physiological stressor that will cause adaptation. And the reason it's imperceptible is we don't feel it. You don't feel the decrease in mitochondrial volume. You don't feel the increased adiposity. You don't feel the loss of muscle mass.
I see. I guess you feel it when you go up the stairs. I've just had
you feel it when you are challenged, yes. But
I just had 7 weeks of not exercising with after doing like a huge, every day for like months and months.
Sure.
And then COVID. It might be partly the COVID, but going up the stairs now, holy crap. Yeah, so I guess I want to clarify a little bit there. So you're saying people don't really think, But isn't sedentary, being sedentary, isn't that considered one of the risk factors for coronary heart disease?
Oh yeah, definitely.
Yeah.
But you don't feel it. I mean,
Yeah.
people often call coronary heart disease the silent killer. You don't feel it happening. You know, when you exercise, you feel the stress because
Sure.
it's high physiological signals that are occurring, or biochemical. When you're inactive, the amplitude of those signals, whether it's in muscle or in brain or wherever, are relatively low. The issue we have is that they're on for many hours, for as long as you're inactive. They're switched on, and causing
I guess
adaptation.
it reminds me in a way of rodent studies, because you're saying often they have a rodent sitting in a cage. It's a bit of a pet gripe of mine. I've brought it up a few times in podcasts. They have a rodent sitting in a cage, and then they'll get it out and run it on a treadmill, and they say that's exercise. But really, that's actually an unphysiological situation in that cage where they don't do any activity at all. So it's extreme sedentary, yeah. And then the stress of the treadmill. So what they can do is obviously put running wheel on there, but then they tend to think of that as training. But thinking about your way of looking at it, it's more like the running wheel maybe is the normally active, and then the not having the running wheel is your sedentary, is your inactive. So you could.
Yeah, I mean, if you look at rodents, their typical daily pattern, they can run, in the region of 10 kilometers a night.
Exactly.
Obviously, they're nocturnal animals, so that, and of course, there are major differences between the rodent and humans in terms of fuel metabolism, and that the rodents' relative metabolic rate is so much higher, so they're far more dependent upon carbohydrate, whereas a human, they'll quite happily chug along, utilizing lipid at all low energy levels.
So I guess what I'm getting at there is that's a way you could, you said it hasn't been looked at in humans, but I guess all those studies that have had rats or mice in a treadmill where they haven't, in a cage where they haven't had a running wheel, that could be your inactive. And then the ones with the running wheel is sort of like your normally active.
Possibly.
Yeah. But then you've got the species.
But my argument would be metabolism in the rodent,
so
in terms of physical activity, is so different to a human.
Exactly.
You fast a human, and their muscle glycogen will remain, as long as they're resting, their muscle glycogen will remain unchanged, relatively unchanged. You do the same to a rodent, and you'll get muscle glycogen depletion even when they're resting. So it's because their relative metabolic rate's so much higher.
So they're very different. So you need to have these studies in humans. So thinking about what has been done, I guess, again, what about these studies that look at, say, insulin? Because I know we're heading towards your immobilization studies that looked at insulin sensitivity. So, glucose uptake in response to a meal or an insulin challenge, etc. What have people done, do you know, with these, studies in humans where they have looked at, I don't know, sitting time and things like that? Is that of any interest?
Yeah, I mean, some, as you'd expect, Scandinavia's led the way. In the 90s, Galbo's group did bed rest studies where they looked at, and the people in that group now are major established figures, Michael Kerr, Fleming Daler, Eric Richter. They looked at leg glucose uptake during a clamp, whole body glucose uptake, liver glucose output. So fantastic research there. And then following up after that, studies out of Copenhagen where they've, reduced step count for 10 to 14 days and saw similar things, induction of insulin resistance, whether you use a clamp or feeding studies. And this is sustained as long as physical activity levels are low. And that's in young and old volunteers. Stu Phillips published a paper with Lee Breen where they took elderly people and reduced their step count to about 1,500 steps per day for 2 weeks, I think. And there they saw insulin resistance developed and suppression of muscle protein synthesis. So it looks like these negative metabolic effects are there whether you use bed rest or whether you use decreased step count or whether you use limb immobilization. It's quite clear. What's interested me is, these studies tended to be pre and post interventions. So, either pre-bed rest and then 10 days later or 2 weeks later. So we've been interested in the speed of onset of these effects, and that's been quite revealing. I mean, I was quite shocked how quickly, if you immobilize an arm from the fingers to the elbow, and you measure glucose uptake across both arms, the non-immobilized arm and the immobilized arm, you'll see a decline in glucose uptake during oral glucose feeding of about 40% within 24 hours in the immobilized arm. So it's specific. It's specific to the limb. So that tells me it resides either at the blood flow level of the limb or within the muscle itself. And then that begs all sorts of interesting questions. So again, studies in Denmark out of Jürgen Wojciechowski's lab and Eric Richter's have done some really nice clamp studies where they've tried to say, well, what's driving this decrease in glucose uptake? And their conclusions were it's probably linked to hexokinase, a decrease in hexokinase activity or glycogen synthase. But I'm beginning to think maybe these responses that they showed after 10 days in bed are actually a consequence of the decrease in glucose uptake, because it seems the decrease in glucose uptake is so fast. And, probably something that resides in the muscle, but something that's detecting the loss of tension rather than linked to, the current targets, which are linked to insulin signaling.
Yeah, so you were saying that, and I put it in the little quote there in the Twitter when I advertised your talk, that you're thinking that the inactivity is not just sort of the opposite of physical activity. Is that what are you thinking there? What's your basis for thinking that?
My basis is the speed of onset is so fast. So as I've just described, you'll get a decline in insulin stimulated glucose uptake within 24 hours of immobilization. You'll also see similar response. You'll see a halving of muscle protein synthesis very, very quickly. So this is a response to the muscle not contracting. So whereas if you undertake exercise, you will get the opposite response. But I'm not convinced it's as fast as, or certainly not, the effect of inactivity is not slower than the effect of exercise. So inactivity per se must be driving these physiological processes. And things like, if you feed someone, for example, when they're immobilized, you get anabolic resistance to the feeding. So the increase in muscle protein synthesis is blunted when you feed someone. Whereas if you feed someone after exercise, the anabolic effect is greater. So there's some very strange
Yes,
events.
that's no, I don't. I guess that's what I don't. Sorry, I'm just trying to get it clear. I guess I don't know why it's, because to me it sounds like, that is the opposite. So when you exercise, you get an increase in insulin stimulated glucose uptake, very like 4 hours. So I've done work with Eric and Jan Wojciechowski as well. So you do the one-legged kicking with one leg, obviously, and then you do a clamp, or we've been doing a meal lately, and you get increased insulin stimulated glucose uptake 4 hours later. And then you're getting a reduction 24 hours later. And then, you're saying, you know what I mean? Like, it is opposite. What I don't get is how it's not opposite.
If you do exercise,
the
effect wanes. The effect disappears.
24 to 48 hours. Yeah. Okay.
As long as you keep someone inactive.
Yes.
The muscle protein synthesis remains suppressed.
Yes.
Insulin stimulated glucose uptake remains low.
Yes.
And actually, another good evidence is that they're different. If you, we've got data, we haven't yet published it, but if you put someone in a cast and then you take them out of the cast and measure, you measure glucose uptake, insulin stimulated glucose uptake while they're in the cast and it's blunted. You then take them out the cast and exercise them. And then you put them back in the cast. The cast can blunt the stimulatory effect of the exercise. So that's telling you the mechanisms have to be different. Okay.
So can I summarize? What part of what I do in these is just sort of summarize where we're at.
Sure.
So yeah, I've been sort of asking, what are the different ways of looking at activity and physical activity, etc. And you're essentially saying that exercise is maybe not just the inverse of a lack of exercise, because exercise has a transient increase in insulin stimulated glucose uptake, definitely 24 to 48 hours or so. But physical inactivity has a continuous reduction. And what you'll get to, which is interesting, is you said you find it very rapidly, and then it doesn't actually change that much. As you get. So why don't you talk about that a bit more, because that's pretty interesting.
Well, I mean, in the paper we've published most recently, what we see is after 4 days bed rest, we have about a 20% reduction in whole body glucose disposal during a clamp compared to before bed rest. And if we maintain those individuals in bed in energy balance for 56 days, there's no change in that magnitude of suppression of insulin stimulated glucose uptake. So it's still blunted by 20% after 56 days. And this is under conditions where, we tightly controlled energy intake. So they're in energy balance. So muscle lipids are not increasing. We measure that intramucosal lipid. So this is a lack of contraction that's driving this rapid decline in glucose disposal, and it doesn't change. What does change is fuel oxidation. So what you see after 56 days is that the insulin-mediated suppression of fat oxidation that you would normally see in a clamp is blunted, which you don't see that at 4 days. So there are metabolic changes occurring in the muscle that favor greater lipid oxidation, but that's dissociated from the glucose uptake effect, which again is quite interesting.
That is interesting.
Why is that happening?
Yeah. So the two studies. So you had the study where you did, just to bring people up. So you mentioned the casting. So you cast them, and then you measured the glucose uptake across the arm, basically. So the arterialized venous. So basically artery versus vein, and the blood flow. Yeah, and it showed that the immobilized one had a 48% or something reduction after 24 hours, and it didn't really change. It stayed like that for the next 3 days, and the immobilized one didn't. And then you found that if you look at, was it 3 days with the bed rest?
Yeah, 3 or 4, I can't remember.
And it was down, and then it didn't change.
Yeah.
So that's really amazing, right? You would have predicted, I guess, it would continue to go down.
And also others have shown, you know, Francis Stevens has shown that if you feed people a high-fat diet while their arm is immobilized, it doesn't blunt the glucose uptake any further. So this is an intramuscular adaptation that is insensitive or not sensitive at all to the circulating substrate availability delivered to the muscle.
I guess it could. I can't help saying this because we did that same one of the studies we did with Eric. We looked at the capillary muscle blood flow. So even though I guess you don't know, because we found that one-legged exercise increased insulin sensitivity partly by increasing insulin signaling and partly by increasing the flow of blood to the muscle. I guess you don't know that,
Yeah, you could. We don't, as I said earlier, we don't really know a lot about the biological mechanisms at the moment. For sure, blood flow could play a role, and measuring microvascular flow is obviously where you should try to be at. All I can say on that is we've got current data where we gave GTN in a deep vein into the forearm to increase.
Sorry, can you just explain the GTN? Sorry.
So GTN donates, I think it's nitric oxide causing vasodilatation of blood flow. So we were able to vasodilate the forearm while it was immobilized and compare it to another arm which we infused saline. And there's clear evidence of vasodilatation. It didn't touch the impaired glucose uptake. But of course, that's I guess, a non-physiological proof of concept. I'm not saying that under physiological conditions, blood flow is not important. Of course, it could be important. And hopefully these as MR methods develop to be able to measure blood flow in vivo at a microvascular level, then these things will hopefully emerge and be clear.
So I guess the nitric oxide, I can't help it again because my nitric oxide is my main area. So nitric oxide is going to see if the smooth muscle's okay. But if you wanted to see endothelial function, you'd have to give something like acetylcholine. So I guess you don't know what's happening to the endothelium. But yeah, it's amazing that 56 days, they didn't get worse in regards to insulin-stimulated glucose uptake. Now, the other thing I noticed was just looking at the paper with the arm, I know it wasn't significant, but if anything, it looked like the glucose uptake might have been higher in the non-immobilized arm, but it wasn't significant. And I was almost wondering, it might be because they compensate, right? You got to use your arm.
Exactly. Yeah.
So it's almost like a positive control almost, that arm went up and that one went down. It wasn't significant.
I mean, it's not in the two studies we published, it's not a significant increase in what we call the control non-immobilized arm. But you're right. Of course, if we immobilize the arm and then we also put them in a sling, so the only arm they have to do things is
exactly
the other arm. So
exactly. So you can't use your other arm either.
we can't tell them not to use that arm. Otherwise,
no, I thought that was quite interesting because if anything, you'd sort of predict that because it's almost showing you that, you know what I mean, that the one they're using became more insulin sensitive, which is what you'd predict. What was the thing I guess was thinking?
The biological mechanisms are as I've said, are unclear. What is clear is that being inactive is not without consequence. People think, oh, I've got to exercise to stimulate adaptation, positive adaptation, which is true. But if you do nothing, you will stimulate negative adaptation. And people don't really think about it.
They don't think through that. You're right. I guess that got me thinking about the participants. It must have been hard to get these participants, especially the ones that lay in bed for 56 days. Did you try and get people that weren't active to start with? Because I guess if they were already active, you'd expect them to have a drop.
No. So we were lucky. So we were able to access the European bed rest facility, European Space Agency bed rest facility in Toulouse. So they have a facility to do chronic bed rest studies. And that's why we had a -6 degrees head down tilt, because it was funded by the European Space Agency as well as the BBSRC here in the UK. So the participants are all recruited by the facility in Toulouse. They tended to be young military males.
Oh, okay. Under orders.
I don't know if they're under orders, but they certainly, you know, were really good volunteers. They understood what was trying to be done, and they, you know, they are well looked after. There's a team with them 24 hours a day. So
I was looking at that, just thinking I would go absolutely nuts. We've done some studies looking at shift work, and we'd have to have the participants, and they could only go out these particular times. They go, oh, I want to go out now and whatever. And I think, wow, compared to you, so your guys were 6%. Now, is that 6% head? So the head is below the, yep.
Yeah.
So 6%. They weren't allowed to sit up, and
no.
they for 56 days. So even going to the toilet, they had to stay in the bed at 6%. I don't know. It just sounds a bit. Do they get claustrophobic? Do they start going nuts? I don't know.
No, I mean, there's lots. You know, we weren't the only study.
Mm-hmm.
Actually, they were in bed for 60 days. It's just our study had to end at 56, so it didn't influence the measures. They wanted to make it 60 days. So that it's a, you know, it's a huge task with groups throughout Europe involved in this one major project.
Tell me, do you get, I've seen a few bits and pieces recently about how exercise increases inflammation, which I don't know if you know about that. That just, to me, it sounds, maybe it's only high intensity. I was thinking, I'm assuming that you'd get inflammatory responses or whatever in a muscle that's sitting there doing nothing, or is that incorrect? Do you know?
Yeah, that's quite a good question. So there's quite a bit of debate going on now about inactivity and then also trauma and inflammation effect on muscle. And people see fairly low level changes in plasma or serum inflammatory markers, which they say these individuals are in an inflamed state, and this can drive atrophy in immobilization. We don't see any inflammatory signatures when we measure gene expression in muscle when people are immobilized, even after 56 days, which contrasts the work we're currently doing, where we're looking at patients who've had ankle fracture and are having surgical fixation. So there you get both a local muscle major inflammatory response in the calf, as you'd expect, and also quite a large systemic inflammatory response. And you also see inflammatory signatures in the contralateral limb in these patients, probably because the systemic response is so large. So there is no doubt, at least in my mind, that, you know, trauma, surgery, infection, all negatively impact upon muscle. I don't believe that is the case for inactivity, because we don't see it in the muscle, and even in the obese, you know, you don't see sarcopenia in the obese. You actually see a greater absolute muscle mass in the obese.
Yes, because they're carrying more weight around.
Yeah.
The other thing I thought about, we did a little bit of stuff. It wasn't mainly me. It was this guy, Edmund Levinger at Victoria University, looking at bone and insulin stimulated glucose uptake. with osteocalcin, which is this sort of bone marker thing. I couldn't help wondering, because I saw some papers there looking at insulin stimulated glucose uptake into bone. So I wonder if it's probably small. But I wonder if, I'm not trying to make your job harder. I'm just saying it's hard to work out what's going on, I guess.
Well, I've put my hands up. I didn't even know there was a measurable rate of glucose uptake into bone. So no comment.
Actually, I'm realizing now, it was actually, that's what it was, because we were interested in exercise, and it was exercise increases glucose uptake into bone. So that was a guy from Finland.
How is glucose transported into bone?
I don't know, but there's a whole bunch going on in there. You know, there's all the medullary area with the storage of fat, and there's I don't know the answer to that, to be honest, but there is a lot of stuff going on in bone.
Sure, yeah, and in inactivity and bed rest. It's just I don't know anything about that.
Fair enough. I was thinking, actually, because when I was thinking about the models, because I was thinking about, you know, choosing different models that we sort of touched on. So, you know, you could do you could get people that are not doing many steps, or you get people that are doing, sitting and then exercising, then sitting the rest of the day or whatever. And then immobilization. It reminded me of something I saw, which is really relevant to what you're saying about, you know, if people have broken their ankles or whatever, that with inactivity, your VO2 max, for example, goes down, you know, across your age as you're aging. And then if you you'll see this like dip, and then it doesn't get back, and it just keeps, then it dips again, you know, and it doesn't get back to where it was. And that was when people were actually in hospital, or, you know, broken a bone or something. So your model is relevant in that regard, of course, as well.
Yeah, I mean, this is a big, quite a common concept in aging research, where you have these, what they call them, are lifelong episodic burdens. You know, whether it's a acute illness and you're in bed at home, as you're getting older, or whether you're hospitalized, you have this period where there's a rapid decline in physiological function, whether it's, you know, in insulin resistance development, whether it's muscle mass loss, whether it's hepatic lipid accumulation. So there's a rapid decline, and it does not return once the person is either, you know, recovered from the illness, or is released from hospital. It does not return to the trajectory it was on prior to the insult, and that's quite common. And we're trying at the moment to look at recovery from bed rest, and trying to see, you know, exactly what does physical activity do after bed rest, and how much do you need? Does it change? How quickly does it change? With that view in mind, you know, clearly, if it doesn't return to the trajectory before illness, that's not good, whether you're old or young. But it could have greater consequences in an elderly person who's already got a low muscle mass, or who is already insulin resistance, or already has fatty liver. So
that's all the more reason.
It's interesting.
It's all the more reason to try and, you know, encourage them to, you know, if you point out to people that, you know, your trajectory is going to be going down quicker, you know, it's all the more reason to try to motivate them to try and do some exercise to get back on that. Hopefully to get back on that.
Sure. I mean, it's interesting. Yeah, you know, it's when you start thinking about it, your head hurts. So if you're training, let's say, so training will increase your rate of muscle protein turnover. So if you then mobilize someone who's been training, let's say they become ill, then the rate of muscle mass loss may actually be quicker, because they're dropping from a higher level.
Mm-hmm.
And you see the same thing in aging studies. So that, you know, people talk about the percentage loss of muscle mass. Let's say in bed rest, they talk about the percentage loss of muscle mass being greater in an older person. But that's, in some studies, it's quite clear that that's because they started off with a lower muscle mass to start with. So when you express it as a percentage of that lower level, of course the mass loss is greater. It's just if you look at absolute mass, the loss is the same. So I guess what I'm saying is even the condition of someone prior to an insult could be important to what happens during the insult.
Yeah, I was actually thinking
there's people working on this idea of prehabilitation, things like this. You know, if someone is going to have elective surgery, for example, the idea is you can actually do something about it before the surgery by exercising.
Just like they've done for years and years with cardiac rehab. You get them fit before you do it. Yeah. So do you have a feel? Actually, I was thinking that there with the muscle mass with bed rest, that's another reason why you'd expect the insulin sensitivity to go down more, right? The longer you're at bed rest, if you've got less muscle.
Yeah, but we normalize the data to mass.
I saw that.
But you're right. You know, losing muscle in itself is not a good thing, irrespective of the effect of the bed rest. But you're right. But the data we have in the bed rest is normalized to lean mass.
Yeah, so I know you said you've started looking at this, but do you have a feel, even from previous literature or whatever, that people who have immobilized bed rest or whatever, whether they do come back quickly? Like, is it like, you know, you lose a certain percentage and then it takes you longer to get it back, or about the same time, or is it not really known yet?
No, I think you can get a return of function. I'm not sure people have looked at, you know, finite end measurements like leg glucose uptake, for example, or muscle protein synthesis and breakdown, how quickly it is, and does it relate to the dose of exercise. I'm sure there will be a relationship, but again, particularly the human studies haven't really been done.
Yeah,
so that's a good area for physiologists to get into.
Yeah,
definitely.
And potential students can contact you. Well, if they're good, that'd be good, right?
Sure.
And actually,
I'm happy to talk to anyone.
yeah. Well, you're talking to me, so you must be happy to talk to anyone. So, thinking about this is something I mentioned to you we sent some notes before we decided before we came on. I mentioned this exercise resistance concept that Eddie Coyle came on the podcast a while back and said he finds that if people sit around all day and then they exercise, they don't get much of an increase in fat oxidation the next day, but he finds if they exercise, then they can make that fat oxidation better the next day, if you know what I mean. So there's sort of this called this exercise resistance. And I thought of that a bit before when you said, I think it was one of the studies you were doing, the mobilization, then you got them to exercise, but then their fat oxidation didn't go up as much, or did I get that wrong? Is that potentially an exercise resistance concept?
No, I mean, you do see it. It's not. So you know, if you're thinking about protein metabolism, you have the concept of anabolic resistance. So if you are immobilized, the stimulatory effect of protein ingestion or muscle contraction on the exercise induced increase in muscle protein synthesis is blunted. So yes, I concur with Eddie that exercise resistance or metabolic resistance to exercise develops if you're inactive. What drives that? We have no idea.
Yeah, so it could be fitting with what you're looking at. Yeah, it could be almost something to add to your arsenal of tests.
I mean, what's really interesting, you know, if you look at, say, muscle protein synthesis in immobilization, whether it's single leg or step count or, you know, bed rest, you see muscle protein synthesis declines, but the pathways regulating muscle protein synthesis during exercise are not markedly influenced. So, you know, if you exercise, the current idea is if you exercise, you'll increase signaling via the mTOR pathway. So the phosphorylation of proteins in that cascade, which switches on muscle protein synthesis. If you immobilize someone, you don't see any detrimental effect on mTOR signaling proteins in terms of phosphorylation, at least, despite muscle protein synthesis going down. So that's further evidence, if you like, that inactivity and exercise are different mechanisms.
Even that reminds me of muscle insulin stimulated glucose uptake as well, because for a while there they couldn't find any difference if you in the exercise leg or the rested leg in insulin signaling. Yeah. But then in the end they found, we found that there was this microvascular flow, and if you looked at other signaling molecules, you could see it. So maybe it's also
likely. It's also likely that, measuring the phosphorylation of a protein may not, or is unlikely to reflect the rate of flux through that pathway. And, so our techniques at the moment are not giving the full picture.
Exactly. And then you would know about some of you said Jan Wojciechowski and Eric Richter, look at the phosphoproteomes. They look at all the phosphorylation sites in the muscle, and there's like thousands and thousands of them. And here we are just looking at one phosphorylation. So yeah, like you say, it's probably a bit more.
So I think phosphorylation in tandem with flux is a good thing, a robust approach, but there are not many people able to do that.
Hey, while you were talking, I couldn't help just looking, I've got my notes here. I meant to mention this earlier, but the incredible effects of inactivity in terms of, being a risk factor for death. So do you want to just flesh that out a little bit for us?
Yeah, not just the risk factor, the largest risk factors. So, there are several large-scale epidemiological studies that are well controlled, some even longitudinal, looking at the death rate or the mortality rate over time, that has related step count to all-cause mortality rates, and also step count to disease-specific mortality rates. Then there's the data of Stephen Blair's, where he's measured cardiorespiratory fitness. So that is probably is a surrogate of physical activity and more accurate probably than step count. And what that work showed was that actually that low cardiorespiratory fitness was a greater risk factor for all-cause mortality in 50,000 people. He sampled 50,000 people in which he measured cardiorespiratory fitness. So the risk factor of low cardiorespiratory fitness was greater than smoking, obesity, high cholesterol, diabetes, and hypertension. So that is phenomenal.
It is phenomenal. Yeah. And you had
people generally don't.
They don't
they don't recognize this.
Yeah, they sort of say, I should be exercising. Well, yeah, so one of the ones you had at the start of one of your papers, large scale, 3,700 men, 1,400 women, followed them for 16 years. There was 450 deaths, and physical inactivity was the central driver of mortality. So that's amazing, because even if you'd asked me that, I would have said, oh, well, cancer, coronary heart disease, at the moment, maybe COVID, but physical inactivity, that's amazing. I mean, I think we all know that.
There's another reason why it's imperceptible. People don't recognize it as a major driver.
When they do, they're dead.
Driver.
Holy mackerel. Okay. So we've got these, I asked people on Twitter if they've got any questions. So we got a few here. We've got Pierre Paquet. He sent through a couple of them we've already touched on. Do you know you probably haven't looked at this, but effective age on these sort of factors? I guess you touched on a bit earlier with bed rest and older people, but do you think if you immobilize something?
Age is associated with greater insulin resistance, sarcopenia, so the loss of muscle mass, age-related diseases. So yes, that there is an effect of age. What is driving this, you know, from a biological mechanisms perspective, focused on muscle, is we don't really know yet. So we know, you know, there's a decline in muscle mass with age. Is this due to cellular senescence? Is it due to, you know, lifestyle factors like positive energy balance, inactivity, or decreasing levels of physical activity? We haven't teased that apart yet.
I mean, I think a lot of it sends, well, a lot of these things where you look at bone mass or, you know, muscle mass or whatever, often it's if people are just inactive, right? They become more less active as they age. And you know, that's the thing. You know, you talk about your models of you can control everything really well. That's the problem, isn't it? Because as people are getting older, they tend to get less active. They tend to get a high percentage of body fat, etc. So
sure, but there's also the way data is being presented, as I mentioned earlier. You know, if you present muscle mass loss as a percentage of pre-bed rest, for example, then if the older person already has a lower mass,
exactly,
then it's going to skew the data. So some people argue, you know, making someone inactive accelerates the rate of muscle mass loss if you're elderly. That's one perspective. The other perspective is elderly people behave the same. It's just that their initial levels are lower.
exactly. And sometimes
some people argue that the rate of loss of mass is less in the elderly, and that may be the case if their initial muscle mass is very low. You know, so if you have someone who's frail, for example, very low muscle mass, putting them in bed may not have a major effect upon their muscle mass because they're already wasted.
Oh yeah, I was wondering, yeah, and sometimes, unfortunately, I think people present the data in the way that supports their argument. Actually, I thought when you were talking earlier, I wonder if, you know how you said with bed rest or immobilization, you get a reduction in insulin sensitivity and then it just plateaus out. I wonder if that's sort of like that gets to sort of like the basal level of insulin stimulated glucose uptake that the muscle sort of needs, and it doesn't sort of have anywhere else to go.
Good question. We're trying to tease that out now by comparing trauma and inflammation plus immobilization post-surgery with just immobilization, because the insult is far greater, obviously, with trauma than just with immobilization. So if it bottoms out, if muscle protein synthesis bottoms out in the way that you're suggesting, then it shouldn't go any further if you have an increased level of physiological insult.
And unless, I guess, it's the cytokine. I guess unless it's the cytokines or something that are making, because is it correct that infection, inflammation, whatever, that reduces insulin sensitivity on its own, doesn't it?
Yeah, sure.
Yeah. So,
Definitely.
right, another one. What have we got here? Oh yeah, I don't know if you know. There's always this question. I ask it sometimes myself. Can you outrun a bad diet? So can a good diet outrun lack of exercise? So I guess what he's saying here, this is Alex. He's got this long French sort of name, but it's in brackets Alex. If you have no choice between eating crap and exercise or eating healthy and being inactive, which option would you choose? It's probably a bit of a weird.
But it's got to be exercise.
Yes.
I mean, if you don't exercise, what you're eating will not really have a major impact upon the endpoint measurements, or relative to the effect of exercise, you know. So definitely exercise.
I tend to agree, but the dietitians won't like us, and that, including my wife.
Well,
Although
I'm not saying there's no effect of diet. Far from. I'm saying, if answering the question, if you had a choice,
yes,
which would you do?
I agree.
Exercise. Yeah.
I actually have to ask my wife. I don't know if you met Kathy, but she's got a master's in exercise physiology and a master's in nutrition and dietetics. So she's unbiased. I think she would probably go with exercise as well, but I have to ask her. All right, Chris Donnelly. Any link between the discussion and the interpretation of studies using rodents in a cage or cells in a dish? I guess we touched on rodents a little bit, but you know, you actually responded, but did you want to flesh it out here? You said it's a big leap between rodents and humans and etc. And cell culture, I guess.
I mean, I think, you know, the rodent isn't a human. I know that sounds obvious, but there's so many differences, particularly when you're looking at inactivity. So, you know, the protein turnover rate of rodent muscle is about 7-fold greater than humans. We know that their rates of carbohydrate oxidation are much higher, both due to their high metabolic rates. We know that the interventions to induce wasting in rodents tend to be more severe than you get in humans. So you know, putting a rodent in a cast produces a more dramatic response than a human, because clearly the rodent doesn't understand.
Exactly. So they get a stress response. Yeah, exactly. All right.
But that's not saying that's not saying rodent studies are not important. I would say that basically if you can show the effects in a human and study metabolism in a human, great, but then that will lead you probably eventually to either rodent or cell-based studies where you try to manipulate your target, you know, which you can clearly do better in rodents and cells. So, for example, in the recent bed rest study, we've postulated that the increase in free calcium as a result of the immobilization, or as a result of the bed rest, is having metabolic effects. So then you could move that into a cell line study where you alter calcium and look at the metabolic effects of that. So exactly. I guess you use the animal models and the cell-based studies to take your understanding from the human studies further.
Exactly right. Yeah. So we do the same stuff. You do as much as you can in humans. If you find an enzyme you think's important, you can knock it out in a mouse. You can't really do that in humans. Although there are some you know, naturally occurring knockouts in humans, but that's a bit harder to find. All right, I think that's about it. Well, actually, one guy here, I'm not sure if you, Ramsey asked, can exercise snacks combat the negative effects of inactivity? How much and how intense? I guess that gets us back to the whole thing about what is inactivity.
Yeah, there's some great epidemiological research in this, looking at associations in huge numbers of people, you know, tens of thousands of people. And there's a really nice study showing, you know, there's a dose-response relationship in terms of intensity. The more intense the exercise, the greater the protection. But you cannot eradicate the risk of inactivity completely by exercise. And what they show actually is quite nice, that even in people exercising intensely, there's a negative effect of sitting watching TV that cannot be removed. So wow. The risk. So it's obviously associative, and you can't, you know, imply cause and effect. But it gives us things, you know, people like yourself and myself who are interested in physiological mechanisms. It gives us a direction where to take research, for sure.
Actually, before we came on, you told me you rode to work today at -3°C. But then you're going to sit around all day and then ride back. So
Yeah.
are you protected or not?
And then sit down again
And watch TV, which is
TV.
a risk factor. There you go.
Sure.
Now, I thought we'll probably finish up pretty soon, but I was wondering if you could flesh out a bit. You said something about you've been looking at the brain, or you've been.
Yeah, we've started doing so that, you know, my career has been focused almost exclusively on muscle and the effects of exercise and inactivity on muscle or nutrition. But what's clear is exercise hits multiple organs. So we're now moving out. Nottingham's really well placed for MR work. You know that Sir Peter Mansfield got the Nobel Prize for inventing MR. So we have.
So we just say
really
you're talking about magnetic resonance.
Yeah.
Just to clarify to people what you're talking
magnetic
about.
resonance spectroscopy and imaging. So we're now trying to look at organ-specific effects of inactivity and exercise. And you know, there's really, again, nice relationships. So there's almost a linear relationship between brain gray matter volume and VO2 max, which is, you know, probably reflecting physical activity levels. And we've done that. What got me into this is we were doing a study of elderly volunteers where we were principally looking at muscle, but we decided to increase the scope of the measurements. So we did brain measurements, and my colleague Sue Francis, who's professor of MR here, she said, "Oh wow, look at this guy, he's got a young brain." That's what she actually said to me. And I said, "What do you mean a young brain?" And then she showed me, you know, the anatomical structures, and sure enough, he was 75-year-old, but highly trained. So we're now doing studies where we're comparing lifelong exercises. So people who've done 100 kilometers a week for most of their adult life versus young volunteers and versus sedentary older people, to try and tease apart the lifelong effects of exercise at a multi-organ level. And that's again, pretty exciting.
It might be that
go back to when I first came on, it comes down to being in the right place around good people. That's, you know, a lot of researchers look.
And keeping your eyes open, you know, looking out for potential collaborators. It might be the other way around. It might be that these people with big brains are smart enough to exercise. So that's the correlation. Because they're smart. So they exercise, so their VO2 max goes hand in hand with it. That's great. The other thing is I've seen bits and pieces, I don't know, but lately I've seen a bit that, you know, doing Sudoku and all these things that are meant to be keeping your brain active is actually been not as promising as they're initially thinking. So they're probably better off exercising for all the other reasons as well.
Exactly. Yeah. I mean, exercise hits multiple organs for sure. And that's really a field that's wide open for, you know, you and I were getting the end of our careers, but for younger people coming into the field, I think being a human physiologist it's really opportune time, because, you know, funding bodies are realizing, you know, animal research hasn't delivered in the way that we'd hoped, and people are now recognizing exercise is indeed hitting multiple organs. So it's a really good time to get into human physiology and exercise physiology, not necessarily related to sport, because, you know, there's not many people that fund sport research, but certainly linked to health and aging. Really, it's a gold.
This has been great, because remember, like 20 years ago, or probably longer again, it was whoever was the head of the American Physiological Society, it might have been Frank or something, wrote an article saying, you know, because everything's got to be molecular biology, and he's saying you still need physiologists to actually work out what's going on, even if you knock out these genes and whatever. But as you're saying, maybe it's going full circle now. You know, you realize.
Yeah, I mean, my personal opinion, and I'm sure that people will disagree with me, but you know, the best biomarkers are only as good as the physiological endpoints that are used to validate those biomarkers. And the better we get at implementing, you know, detailed, dynamic physiological measurements in human volunteers, the better biomarker approaches will be, and the better research discovery will be.
That makes sense.
So physiology needs to be at the center.
And in terms of, I think this is something that's come out in lots of podcasts, is how exercise is affecting different organs. So, you know, I had John Tifo on talking about the liver. You know, we've talked about all sorts of things. And I've actually tried to get Mark Tarnopolsky, and I've asked Mark Fabreo to come out, because, you know, these exocrines that are, and exosomes that, you know, are getting released from the muscle and traveling all over the body. So it's really like, as you say, it's a massive area to try and look at this. And it's integrative physiology at its best.
Exactly.
Yeah.
And it's really good for young people coming through. You know, everyone, certainly in the UK, times are tough in terms of the economy and research funding, but this is really, I think, something to get excited about. And you know, hopefully young people will, you know, training in human integrative physiology will increase.
Exactly.
That's important.
Exactly. All right. Well, thanks a lot. Now, what I might just do before we finish up is just if you can tell us a few sort of takeaway messages. So if you wanted people to have 2 or 3 things they take away with them from this chat.
Okay. So, well, firstly, I think inactivity in itself is a physiological stressor. So it will, you know, exercise will produce positive effects. Doing nothing will produce negative effects. So that's the first thing. The second thing is that, you know, these effects are very rapid with inactivity. So within 24 hours of being inactive, you will see, or immobilized, you'll see impaired glucose disposal and muscle protein synthesis declining. And the third take-home message is epidemiology has, you know, provided a lot of robust associations between physical activity and health and mortality rates, but the biological mechanisms that are driving these associations are not yet clear. And this is therefore a really good time for exercise physiologists and metabolic physiologists to get involved.
Totally agree. Now, one thing I've realized I've not brought up, because we've had so many people come on and talk about diabetes and things, is I guess we haven't really said why is it important that, you know, if you have a lower insulin sensitivity. You know, did you want to just flesh that out a little bit just to finish up? Something we should have said at the start, probably. Yeah.
Well, insulin resistance is associated with negative metabolic effects that actually, you know, progress and become even more pathophysiological as time passes on. So, you know, you have endothelial dysfunction, you have cardiovascular cardiac dysfunction, you have usually in tandem accumulation of lipid within muscle, and that's both, you know, between muscles and within the muscle fibers themselves, all of which alter physiological function and actually probably accelerate the rate of further metabolic decline or further insulin resistance developing.
And you can end up with, you know, pre-diabetes and then diabetes and then everything that goes with that as well.
Yeah.
All right.
Sure.
Well, thank you very much.
It's a gradual decay from a healthy state into a pathological state, you know, and they go hand in hand with other diseases, multimorbidity, you know. So the common drivers of multimorbidity, which is the occurrence of one or more chronic diseases are sedentary behavior, excess energy balance, age. So everything we've been talking about.
Mm-hmm. And even insulin resistance increases your chance of cancer as well. So yes, that's very important. And as you said at the start, and we'll finish again, it is imperceived, which is a word I actually, to be honest, had to Google. It makes sense. It's the opposite of perceiving it, but it wasn't one I was familiar with. So it sneaks up on us. All right. Well, thank you.
It's silent. It's silent.
Silent. Yes. Okay. Well, thanks again, Paul, for coming on.
Okay.
I hope you don't get too cold riding home later on. Okay.
Yeah, it looks nice and sunny where you are.
It was 37 today, which the Americans will think, oh, that's freezing, but 37 Celsius is 38 is 100 degrees Fahrenheit. Okay, good on you, mate. Thanks again for coming on.
Thanks, Glenn. Bye.
Okay, bye-bye. I hope you enjoyed this podcast. Please like, subscribe, pass it on to your friends and colleagues. Check out the other podcasts. Thanks again.